everyone. Good morning or good afternoon, depending on where you are joining us from. It's a great honor, great pleasure to welcome you all to this seventh day of the 2026 ICDP School on Synchrotron, light sources and their applications. So my name is Mariel Agbangbata. I'm chemist and the coordinator of ESTEC Lab in Benin Republic. With a great pleasure, I will be sharing this session today with the help of Dr. Samsam from Italy. So our first topic today is titled Macromolecular Crystallography in Cancer Biology, and it will be presented by Dr. Silvia Onesti from Elettra Synchrotron in Trieste, Italy. I will give a very short introduction about Dr. Onesti's work. So she is graduated in chemistry from the University of Pavia, followed by a PhD in structural biology from Imperial College in London. She worked at Imperial College for more than 20 years at the interface between the physics and the life science department. Since 2009, she has returned to Italy, where she founded the structural biology laboratory at Elettra Synchrotron in Trieste. So her research focuses on understanding the molecular aspect of proteins involved in DNA replication and the maintenance of genome stability. All these using an interdisciplinary approach that ranges from biology to physical techniques such as X-ray diffraction and electron microscopy. She has organized conferences and workshops in Italy, UK, Canada, India, South Africa, Senegal, Tanzania, and Rwanda. And I should mention that she was also in Benin at ESTEC Lab. We were very happy to welcome her. She has been a member of the International Union of Crystallography Commission for Biological Macromolecules and was recently elected a member of the Academia Europea. Thank you very much, Dr. Onesti, for joining us today. And I'm very honored to share this session. And now I will give you the floor. You can start sharing your screen.
Thank you very much for this very kind introduction. Let me just start with now. As always, in this kind of presentation, one has to sort of touch upon things because it's too difficult to go very much into detail. But what I'll try to do is to give you an idea of how macromolecular crystallography can be different from small molecule crystallography, how it uses synchrotron radiation, but more than that, the development that are going on in structural biology. And I'll just give a brief example of my own work. So I'll focus more on the technique than actually my research. So just to start stressing how important has been crystallography and structural biology in the development of life sciences, I want to stress that humans are very, very much visual creatures, and often we use our eyes to really understand the world, and we often think that we understand something when we see it. That's why it is very important to draw diagrams, to see sketches, and so on. And quite, rightly, Richard Feynman, in his famous lectures in 1959, was discussing how much there is still to discover, and he stressed quite a lot the development that were required in biology, asking which are the most central and fundamental problems in biology today, which are the most important questions we can ask, and he replied that it is very easy to answer many of these fundamental biological questions. We just need to look at the thing. And really, this is what structural biology has been doing. The idea is to look at biological objects in great enough details to understand, to visualize atoms, and therefore to be able to understand the chemistry of these molecules, and therefore to be able to understand all the components of these complex molecular processes and their complex mechanism of action. And indeed, structural biology has been massively contributed to major biological discoveries and has very much facilitated also the development of therapeutic agents to cure diseases. And I'll tell you very, very briefly something about cancer biology, but Tandeka, that is following me, will discuss the role of structural biology in infectious diseases. So why structural biology? Well, beside understanding biological objects at the molecular level and therefore making us understanding better biology, it provides an important framework for rational structure-based drug design because it provides models that can be used for virtual screening, models of proteins and inhibitors that can be used for the optimization, for the rational optimization of lead compounds, and also development like fragment-based drug design. So as an example, which is a pretty old example, this is a protein kinase that is bound to a drug against leukemia. Just to give you a diagram that you've seen probably multiple times, here I drew a number of important biological molecules, and here are the techniques that you can use to actually visualize this object. And mostly in structural biology, we are within this realm, and therefore we visualize nucleic acid, proteins, proteins embedded in the membrane, but also macromolecular complexes like ribosome, also viruses, and some structures within, for example, the cell membranes or within the cell in cryo-electron tomography. So I'll give you a brief overview of macromolecular crystallography that relies on obtaining well-diffracting three-dimensional crystals of the macromolecule we are interested in and studying it using X-rays. So this is the typical pipeline of a macromolecular crystallography experiment. And as you can see, you span a variety of techniques that go from biology to physics and bioinformatics and data science. We typically start from selecting a target protein using the gene and using molecular biology to clone the gene, express and purify soluble protein, and that's often one of the bottlenecks that you may encounter. And then major bottleneck is to use, try to sort of force this soluble protein into well-diffracting crystals. Once you have crystals, you can collect data, typically using synchrotron radiation, and then in a way that is a bit more complicated than what happens with canonical small molecule structures, you can actually guess the phases, build your electron density that you interpret in terms of a molecular model, obtaining a structure, and this structure can then be validated, but also used for following up bioinformatic analysis and, for example, to design potential drug leads, inhibitors, or interpret the results from the biology. Now, one of the major questions when macromolecular crystallography started is whether crystallization affects the structure. People were, more confident with small molecules, but with large and flexible macromolecules, there was quite a lot of doubt as to whether the structure we see in the crystal does actually have any biological sense. And first of all, there is a theoretical argument that is that protein crystals are very weak. They're held together by just a handful of hydrogen bonds. So the forces that hold the molecules in a lattice are much weaker than those that holds the whole protein structure together. So it's unlikely that the gaining of a few hydrogen bonds could completely modify the crystal, the protein folding, which requires thousands and thousands of hydrogen bonds and other molecular interaction. But more than that is the fact that experimentally we know that many proteins which can be crystallized in different crystal forms and therefore with very different lattices are often identical or almost identical. And more than that, it was already observed many years ago that some enzymes can actually retain their biological activity within the crystal. And that's a strong indication of a native-like structure. I'll show you one example. Sorry, these are still the hydrogen bonds holding crystal together. But this is an example from my own work. I worked on an amino acid tRNA synthetase, which is the key enzyme that translates the genetic code because it connects the correct amino acid with the correct tRNA that translate the genetic code. And the first step of this reaction is joining the amino acid, and here you can see the amino acid lysine, with ATP to form a lysyl adenylate. And when I use the non-hydrolyzable ATP analog, I obtained in the active site of my protein this electron density, where you can see the two molecules clearly separated. But when I actually used the real ATP within the crystal, I could form the lysyl adenylate and break the bond with the pyrophosphate, which is the living group of this reaction. So we know that the structure we see can actually have some enzymatic activity also within the crystal. This doesn't happen all the case, but it has been seen in a number of cases, giving us confidence that we are really looking at crystal structures which are the structure of the protein in solution. There are a number of other techniques and cryo-electron microscopy that I'll touch upon in the second half of my talk, which also provide now atomic structure of proteins, and in most cases the structures are virtually identical to the one determined by crystallography. However, we need to remember that a crystal lattice may favor a conformation that not always is the dominant shape in solution. So it's always very good to double check our results using other technique, like, for example, small angle X-ray scattering, or, for example, electron microscopy. As you all know, in crystallography, there is a phase problem, and the phase problem that has been easily solved for small molecule, but in macromolecular crystallography, we are facing a problem which is more complex, and that's because small molecule crystals have a small unit cell Therefore, you have a coarse sampling, a large a pattern with a large a reciprocal lattice, which give us a few very strong reflection, and typically small molecule crystals diffract to very high resolution. While for protein crystals, we typically have large molecules, large unit cells, smaller reciprocal lattices, a lot of very weak spots, and many weak reflections that diffract to low resolution. So we have, rather than having to face a handful of reflection, we need to face 10,000, 100,000 reflection, and we are also facing a situation where we cannot assume very high resolution. So as you know, the small molecule problem, the phase problem in small molecule is these days solved by direct methods, applying physical constraints such as positivity. The electron density always has to be positive, atomicity, the electron density that I obtain at the end has to come from single atoms, and so on. In such a way as to limit the phases to one of a few possible self-consistent sets. In macromolecular crystallography, we have a much larger number of reflection, and especially we typically have data at lower resolution, so a strong constraints like atomicity does not hold. And therefore, we need to use experimental technique to solve the phase problem in macromolecular crystallography, and the three main techniques that are used are multiple isomorphous replacement. This is the older method that was developed by Max Peruzza with co-workers like Francis Crick and David Blow, and I've been fortunate enough that David Blow was my PhD supervisor, so I heard firsthand a lot of the stories of how they were working in Cambridge, and this relies on binding to the macromolecular crystals a handful of heavy atoms and compare the diffraction pattern of the protein heavy atom complex with that of the native. It's a technique which is a bit time consuming because one has to do a trial and error search for good heavy atoms that binds to the crystal, maintaining the isomorphism and therefore allowing us to really compare the data. Whenever it is possible, and that's a technique that has been developed in the 70s, 80s, I can actually exploit the knowledge of the existing, of the similarity of an existing structure, and this technique is based on the theoretical work of David Blow that we mentioned before and Michael Rossmann, but has been then fully developed by Michael Rossmann, and relies on the fact that if I've got a homologous structure and I have a sequence similarity that allows me to believe that the structure is likely to be similar, I can exploit the information of the similar structure to determine my some initial phases for my new unknown structure. This technique has also, until very recently, could only be used if there was an experimental structure with a high sequence homology, but the new development in artificial intelligence-based prediction now allow us to use these prediction, which not always work, but in some cases they do work. And I have a couple of slides on how artificial intelligence really is changing the landscape of structural biology. The experimental technique that is mostly used these days relies heavily on synchrotron radiation and on the absorption of specific wavelengths that trigger electronic transition within the atom core. Is similar, the formalism is very similar to MIR, but it gives in general more accurate phases and it's quicker when it works. However, it requires specialized synchrotron beamlines with small bandwidth and that are tunable, so I cannot use them on the traditional X-ray generators because I cannot control the wavelengths, and also on the presence of anomalous scatterers. And one of the tricks that we often use is to produce the recombinant protein using selenomethionine instead of methionine, and therefore labeling intrinsically the protein with selenium atoms, which have a very convenient absorption edge, which is close to 1 angstrom. Now, of course, in a 1 hour lecture, I don't have the time to go into details into this technique, but I just want you to know which is the general framework. As we mentioned, collecting data is very important, and synchrotron radiation has made a remarkable difference to structural biology, because whereas in laboratory sources I need low intensity, long exposure times, and these trigger a much more serious problem of due to radiation damage, because proteins are labile. With synchrotron sources, I can have a tunable source, changing the wavelength, and that allows phasing using MAD, as I just mentioned. But also the high intensity means that I can use much shorter exposure times, and I can therefore have less damage, because the damage is not only due to the primary damage and therefore the number of photons, but also to the formation of free radicals. And the longer is the experiment, the longer is the time for the radicals to cause further damage. So most protein crystals are small, diffract poorly, and only the intense X-rays from a synchrotron have allowed to determine structures of very large and very complex proteins of medical and pharmacological interest. The good news is that these days you don't even need to travel to a synchrotron to do crystallography. Most macromolecular crystallography synchrotron beamlines are highly automated, and they all, or most of them, offer remote data collection, and it's remote data collection that can be used by both expert user, but there is enough support to allow also novices to use it. This is the setup of what was until very recently the current macromolecular crystallography beamline at Elettra. And Maurizio Polentarutti, Giorgio Bais, and Nicola Dimitri are the current member of the beamline. Annie Heureux and sorry, I forgot his name. This is a postdoc that has left a few years ago. So, and Baba Mangiasetti as well are not here. There is another Indian postdoc. This is a beamline that has been built with a major contribution from the Indian Institute of Science, and therefore is an Indian that it is shared with the Indian structural biology community. And we still hope, together with the Indians, we are in the process of building a much more advanced beamline that will allow the possibility of doing microfocus crystallography, of using a much smaller crystal and having a much better collimated and more intense beam. So if you, I mean, as you've heard probably already, Elettra is undergoing a major refurbishment, and in about a year, we hope we'll be able to host both, in-person users, but also remote users to do this kind of experiments. So once we have collected data, somehow determine the phases, we can build we can calculate an electron density map from which we can actually build a model. I have an interest in the history of science, and I like to remind younger people how complex it was to do some of the things that now we give for granted at the very beginning of this technique. I actually never really had to build a model like that, but I was involved. I was at Imperial College, which is next to the Science Museum, and for one of their exhibition, me and a couple of other PhD students were actually paid by the museum to rebuild a model like that in the museum. And I think this is the hardest earned money I've ever obtained. It was actually a lot of work using this kind of map and what is known as the Richard box. It's a complex setup that allowed, building reasonably accurate models in the 60s. Now, of course, we use high-resolution computer graphics. And what I want to stress is that these programs are very easy to use, but it actually, the kind of results you get, it depends critically on the resolution you manage to obtain. And if we have a very high resolution map, building a model is a bit like joining the dots in a enigmistic game. But when we have low resolution maps, it's much more difficult to identify the features, and we do have a number of important structures which are halfway between these two situations. So where we have models which are reasonably accurate, but not as accurate as atomic resolution maps. So there is quite a lot of gray area in structural biology. So again, if we visualize the pipeline, we need to purify the protein, crystallize it, collect diffraction data, process the data, somehow solve the phase problem with one of these techniques, MIR, MAD, molecular replacement, obtain an electron density map, and then refine our model, and finally validate the structure and deposit the model in the protein data bank. However, because structural biology has been so successful, crystallographers started to tackle larger and more complex structures. So the problem we need to face is that some proteins have multiple structures with multiple domains connected by flexible links, and also they can be part of very complex macromolecular machines. So the textbook pictures that we had of the cell like a soup where each single protein and enzyme swims freely in the cytoplasm has really dramatically changed. So cells are very crowded environment and macromolecules interact with each other in complex and dynamic ways. So we need to integrate multiple approaches, including macromolecular crystallography, but also SACS, cryo-EM, NMR, computation, single molecule studies, and other biophysical techniques to really get a picture of what's happening in life. I'm just going to give you a very brief description of cryo-EM because really it's now become one of the essential tools for a structural biologist, and most synchrotron have a cryo-EM instrument that they treat a bit like a additional beamline, and very often the kind of sample we need to use can actually be tested both by macromolecular crystallography and cryo-EM, and you can also use a microscope to do to analyze very small crystals that are too small for X-rays. So there is a continuous crosstalk between structural biology techniques. So in single particle electron microscopy, you still have to purify your protein, but then you actually freeze your protein and look at the frozen layers of molecules with a transmission electron microscopy. And you have a picture like that where you have your molecule in all the possible, ideally, in all the possible different orientation. You can then pick the particles. Typically, a micrograph doesn't look as good. This is obviously a teaching tool, and actually I need to acknowledge that is a slide from Helen Seibil from London, and she's one of the pioneers of this technique. And here is where it gets tricky, because you have to use very low doses. It's very difficult to get a signal from, you get very noisy images, and you have the problem of getting a signal from very noisy images. But what you can do with your particle is classify them, putting together the picture of the molecule that has ideally the same orientation. So you put them in classes, and then you average these classes, getting class averages where the noise is cancelled and the signal gets enhanced. And then you use these classes to build a 3D reconstruction from this 2D projection, and then you go back, and where initially you classify more roughly, if you want, whether you're looking at a molecule from the top orientation of 5 degrees twist, you don't care, but the more you go ahead with the refinement, the more you subdivide the classes, and you get higher and higher resolution. One of the advantages of cryo-EM is that obviously your biological samples, which is often a complex molecule that can assume a different conformation, so may be present in this conformation or this. But you can have also molecules that are like that, which are in a really bad shape. And one of the advantages of cryo-EM is that you can do in silico purification. So you can, obviously, in the lucky case, disentangle multiple conformational states and obtain 3D reconstruction, separate 3D reconstruction from the 2 or 3 conformation that you have in solution. Of course, if you have many different conformation, it is a problem, but sometimes you manage also to look at a continuum of conformation and try to detect particular movements which your sample, your micrograph may sample. Now, I've done some cryo-electron microscopy when I was in London, but at that time it was a very low resolution technique. But about 10 years ago, there has been a revolution with a new generation of detectors with unprecedented speed and sensitivity that allowed the use of lower doses, therefore less radiation damage, less noise, and therefore providing higher resolution data, but also with a much faster readout that allow to compensate the small movement that happen when the electron beam strikes the sample and makes it vibrate. And this has generated a resolution revolution that provides structure at higher and higher resolution. We can now, the top laboratories in the world can see single atoms, but also it allows to see smaller and smaller molecules. When I was in London, you couldn't dream to do molecules which were larger, which were smaller than 300 kilodalton, and now you can do smaller molecules that are much smaller. However, we need to remember everybody that this is not a magic technique. One still needs to do a lot of work, produce recombinant protein, whereas one can get to atomic resolution. This is very, very difficult and very hard, and the majority of the cryo-EM structures that are deposited in the protein data bank are around 3 angstrom resolution. So we are halfway between the kind of resolution I show you. Anything below 150 kilodalton is still rather difficult. 100 kilodalton molecules can be solved, but they require quite a lot of work. And in general, collecting good data often requires quite a lot of work, optimizing the conditions to avoid the damage at the air-water interface, to find the optimal freezing condition, and so on. And at 3 angstrom, drug design is possible, but is still not ideal. So just to stress that crystal structures still have an important role to play in structural biology. Despite that, crystallographers are sort of becoming a bit more dismissive towards diffraction pattern, and they are looking with a lot of desire at electron microscopy. And indeed, we've done the same. After a long campaign, we finally managed to get a new cryo-EM facility, which is being established right now, which includes one of the top-of-the-range microscope, not really the top of the range, but still a microscope that allows to do reasonably medium high resolution structure. And the idea is to use it not only to do single particle analysis, but we always also have a Fib SEM that will allow to use it for cryo-electron tomography, and also will be able to use it with small crystals to do micro electron diffractions. But all this is in the future, as the microscope is just now being tested. I want to spend just a few minutes to discuss, where structural biology is going. And one of the things you all know is that there has been an incredible development in the prediction of protein structures. And we obviously exploit this development because one of the first things we need to do is to do a bioinformatic analysis to identify the part of the protein we really want to express. Very often, if you remember my protein made up of flexible domain, we also try to express the full-length protein, but often what crystallizes and work is just a folded compact domain. So AI prediction allow us to better define the domain boundaries to produce protein. They obviously provide models to obtain phases by molecular replacement. I told you that these days, while before for molecular replacement, you needed an experimental homologous structures. Now you can take also the AlphaFold, Rosetta fold model and try it. They provide also model to fit medium low resolution cryo-EM, cryo-ET maps, and of course provide models for computational biology and can be used also to design more stable mutants and so on. However, there are limitations. This technique works much better with single protein and domains. A lot of biology involves complexes, and although the latest version of AlphaFold is becoming better at predicting protein-protein interaction, for example, in my experience, protein nucleic acid interactions is still far less accurate. Also, typically, the models do not include ligands or cofactors. Some cofactors can be inserted in AlphaFold 3. They don't include post-translational modification that sometimes are very important. Moreover, I think the most important point is that proteins are not static or rigid assembly. They exist in multiple conformations, and often a crystallographer goes on for many, many years to look at all the possible conformation at the protein in the presence of the substrate, an inhibitor, the product, and looking at all the gamut of conformation that really allow us to understand the chemical reaction at the molecular level. While these algorithms give one conformation that may be more or less right, often is reasonably good, but is not always the whole story. And as I mentioned before, drug design tends to require better precision. I'm now going to spend a few minutes to tell you briefly what I work on, and my favorite enzymes over the last 10-15 years have been helicases. Helicases are ubiquitous enzymes, and as the word say, they deal with the DNA double helix, and they're true molecular machines that use energy from ATP hydrolysis to walk along the nucleic acid and open or remodel the nucleic acid. They are found everywhere in life, from viruses to bacteria, archaea and eukaryotic cells. Being essential in nucleic acid metabolism, they are involved in a variety of nuclear processes like DNA replication, because before copying the DNA double helix, you need to open it up, but also telomere maintenance, segregation of chromosome, transport in and out of the nucleus, biosynthesis of ribosome, translation, splicing, maturation of RNA, transcription, and DNA repair. And due to their importance in the cell, they are emerging as a new class of antibacterial, antiviral, and anti-cancer drug targets. And I'm focusing especially on helicases involved in cancer biology because they have a very important and complex role in tumor development. First of all, often helicases are actually overexpressing cancer cells and are therefore proliferation markers, so they can be used for diagnostic, but also being essential in some cellular process, they can be used as a target for anti-tumor drugs. Because you can design small molecules that interfere and they stop cell proliferation. And they also can be involved in the resistance of cell to radio or chemotherapy. So designing inhibitors of some of these helicases can help making the current therapy more effective. And because I don't have a lot of time, I'm really going to take only a few minutes. I'm just going to stressing one of our projects. We are interested in a number of helicases which have an iron sulfur cluster. They're also present in pathogens, but they are mostly eukaryotic cells. And in particular, we focus on DDX11 and DARTL1. I'm only going to tell you about DDX11, which is essential for the separation of chromosome during mitosis, and it's essential for DNA replication and DNA repair. And in collaboration with Solaris, we have first of all purified in our lab the protein. You can see that is yellow because it has an iron sulfur cluster. But we've been using cryo-electron microscopy to determine the structure, and the first structure we obtained was at about 5 angstrom resolution, but even at this resolution, you can clearly see this is the AlphaFold model, and you can see reasonably well how the DNA binds to this protein. And if we take the difference between the density we obtain and the AlphaFold model, we could clearly see both the double-stranded, but also the single-stranded DNA bound to the protein. And one of the things I want to stress, and the reason why I choose this protein rather than one of the crystallographic protein, is that I want to stress that the AlphaFold model predicts a very long, this insertion to be folded into a very, very long helix, which has a reasonable degree of accuracy. I mean, this part is predicted as not very accurate, but this light blue part has a reasonable degree of accuracy, but you can see that there is not a hint of electron density here. We solved the multiple structure of the protein with the double-stranded DNA, without, with substrate like G quadruplexes, and we never have this electron density. So this is just to stress that we still need experimental structure despite the progress of artificial intelligence-based prediction. And actually, we now have a 3 angstrom structure resolution, so we are close to the kind of structure you obtain for crystallography, and this is very much work in progress. We are currently building this model. And I mean, these are a number of people which are working in my lab, and the funding I obtained, but I want to take the last few minutes. I'll actually skip these 2 slides because I don't want to take too much of your time. I might go back if you tell me that there is enough time, and I want to use a couple of minutes for 2 advertisements.
3 minutes left. Thank you. 3 minutes left.
Okay.
3 minutes. Thank
All
you.
right. Now, I want to first advertise a conference I'm organizing together with Annalisa Pastore and Gian Gaetano Tartaglia. And it's really a conference on the interplay between structural biology and machine learning and their role in de novo protein design and drug design. And one of the things I want to stress is that we are openly trying to address the relevance that this new development have in emerging countries, and therefore we have a program that involves scientists from Asia, Aravind Penmazza, Africa, Trevor Sewell, that some of you know very well, and from South America, Richard Garratt, that also some of you know very well, really with the aim of discussing how this new development affect the whole world, not just the Western world. And the second advert is that as part of the ICTP program of conferences, we've been organizing a number of biophysics schools. And in the past, we had biophysics school in South Africa, Senegal, Tanzania, Rwanda, and the next one will be in December in Addis Abeba. The organization is actually includes also myself, but is mostly done by our Ethiopian colleagues. And therefore, for anyone that is interested in biophysics, we cover both structural biology, but also nanobiotechnology and computational biology, if you just have a look at the ICTP program. And I'm just, I think I will stop. I can leave this slide here just to see if there is anyone that has questions or wants to focus more on these things, but I'd rather stop here than go on until, you know, the time the next speaker
Yeah. Thank you very much. Yeah, thank you very much for the interesting talk and especially for the opportunities that you share with us. Thank you. So we'll start with the questions, and I have some in the chat. So the first question is from
the use
okay,
of
Sonia. Sorry.
Now I'm looking. Sorry.
Yes, I would like to read you the question from the chat.
Okay, let me look at
so the first one. Yes, the first one is given the progress in cryo-EM, does crystallography and diffraction still have anything to offer in the Prof. Onesti's field?
Yes, I mean, certainly, I mean, as I mentioned, cryo-EM is certainly a very, very useful technique, but when you manage to get crystals, you typically have more accurate structures. So it certainly is very useful. And also, once you have crystals, it's a lot easier to see a lot of different protein substrates to screen quickly protein substrates. So if you can get crystals, certainly this is really very useful. I mean, just to, you know, when I thought about future perspective and the possibility of going back to X-rays, and you may be able to still see my screen, certainly there are a number of development in the use of time-resolved experiments, which are not easy. They've never been very easy to do because it's not easy to trigger the reaction inside crystals altogether, but there are there is still quite a lot of work that can be done. So I don't think that crystallography is at the end of the line. And I think it has still a role among the technique. It has probably lost the crown of being the queen that has been for a good 70 years. So, but
Okay.
I think you can easily use both. Once you've made the effort of producing your protein.
Yeah. Thank you. So there are very interesting comments in the chat. So thank you all for your comments. Another question is this. With regard to possible sample damage from laboratory source versus synchrotron source, aren't high brightness and short exposure time versus low brightness and long exposure time almost practically equivalent?
Now, no, because what's happening when you expose, to X-rays is that you've got a primary damage, which is the damage caused by the X-ray photon. But then what happens is that you form free radicals. And these free radicals trigger chain reaction that cause more damage. So that's one of the reasons why you freeze crystals. You freeze crystals because you stop the free radicals from freely moving through your sample, but also it is much better to just give a lot of photons very quickly than the same dose over a much longer time. And that's because the major damage is due to the reaction, the free radical reactions triggered by the initial damage.
Okay, thank you. I think we can take one more question before moving to the next speaker. So this one is, can we think of the holographic methods using a kind of reference to visualize these macromolecules?
Now, X-ray absorption spectroscopy, now it would be technically, very complicated to do. To be honest, this is outside my field of expertise, so I don't want to say things which are not, but you have to think about the complexity of macromolecular structures. So you are not recording just one image, but quite a lot of images. And the holographic method, your phase problem is not your major problem. If you get good diffracting crystals, you solve the structure. So one way or the other, it takes you a bit of work, but one way or the other, you do solve the structure. As for X-ray absorption spectroscopy, it can certainly be used, and it's used, for example, in XF experiments when you have, that's one of the things that I want to do with my iron sulfur cluster, for example, to really try to look at, the exact chemistry of the cluster. So in general, I would say, besides structural myology, there are a number of biophysical techniques that can provide important information.
Okay. So this one is from Hamed. What about having a school on cryo-electron microscopy and structural biology in Egypt?
I would be delighted to help to organize one.
Okay. So, why are gross conformational changes unlikely during
Sorry?
protein crystallization?
What are gross conformational changes? Why are gross conformational changes? I mean, you could have conformational changes that may also, let's say you've got a molecule that has two different conformation, a closed and an open one. You may have that in solution, 80% of your molecules are open and 20% closed, but the closed conformation is easier to crystallize. So you would get a crystal structure that gives you only one of the conformation. But when I say that, it's very unlikely that the process of crystallization manages to disrupt the conformation that the protein naturally has. And that's because when you think about the protein crystals are extremely fragile. If you touch one, it shatters. And that's because typically you've got, I don't know if you can see your hand, I don't see myself anymore. So I hope you see my hand. And maybe if I stop share, it'll be easier.
Yes, we can see your hand. We can
So
see your hand.
yeah, you have one molecule here, and this makes 3 or 4 hydrogen bonds with a molecule here, and 3 or 4 hydrogen bonds with the molecule there. So protein crystal contains a lot of solvent, which is good because we can soak in is substrates and inhibitors, and you can um, you have the molecule that is almost at the liquid state. So gaining the energy that is provided by these 3 or 4 hydrogen bonds is not going to be enough to undo the energy minimum that the folded protein is in. This is what I was trying to say. Not sure if I made it clear. But
Yeah.
I mean, but
Okay.
this is only the theoretical arguments. In practice, we do have multiple copies of the same structure. For example, we solved, polymerase beta from one organism, another one from the same organism in different conformation, and we can compare a lot of structures and see that they are virtually the same.
Okay. So, another one, what is the phase problem in macromolecular crystallography?
Okay, this is a problem in all crystallography, and I was giving for granted that you've been already taught about this, but I'll tell you very, very hand waving briefly. When you shoot X-rays onto a sample, you have the electrons in the sample that scatter the X-rays, and then you collect the scattered rays onto a detector. And the scattered rays are electromagnetic radiation, which have an amplitude and a phase. And what's happening is that you measure how black is the spot, and that gives you the intensity. I mean, you measure the intensity, take the square root, and that gives you the amplitude of the diffracted beam. But you lost the information on with whom which each of the diffracted beam, the phase with which the diffracted beam have one with respect to the other. And this information is essential to then use this data to reconstruct an electron density map. You can show, and you can show this mathematically, that the electron density map, which is the blue wire I show you, is the Fourier transform of the diffraction pattern. But the diffraction pattern includes both the amplitude and the phase. and you lost the information about the phase, which you need to somehow recover. I'm sorry, it's very difficult to explain it in a couple of minutes, hand waving. But if Zahid, if you write me an email, I can send you slides from my course where I dwell on that more in details.
Okay.
Thank you so much, Silvia.
I can put here my email if anyone has a
Yes.
question.
Yeah. So we can stop here.
Yes, please.